Welcome you all to this cryo-EM course. Last class we discussed about that high pressure freezing or self pressurized rapid freezing, and that time I also talk about that cryo-electron microscopy of vitreous section, how we can do that cryo-electron microscopy of vitreous sectioning, that we are going to discuss now. In order to image bulky specimens at the cell or the tissue level in the transmission electron microscope, sample has to be either thinned by focused ion beam or mechanically sectioned. So, there is a two way that we have to pay attention, that either if the specimen is very thick and then we want to go inside that and we want to image the cell. What is important, that first we have, because any cell it has a cell membrane. So, it may be a plasma membrane and or the any kind of biomembrane. So, different kind of lipid layer it will be there. So, we have to rupture that lipid membrane. When you will rupture that lipid membrane, we can see inside the cell. So, there are different way to do it. So, one way it is that either we can use that FIB SEM, that is called focused ion beam, FIB SEM, that is focused ion beam, or mechanical section. So, mechanical section you can use cryo ultra microtome, or you can do the fridge fracture. If you do the fridge fracture, you can also break the membrane bilayer and then you can see inside that. So, here today the latest technique that we are telling, that cryo electron microscopy of vitreous sectioning, that is called CMOVIS, employs a cryo ultra microtomy to produce sections with the thickness of 40 to 100 micrometer of vitreous biological materials suitable for cryo-EM. So, CMOVIS consists of trimming and sectioning of the sample with a diamond knife or maybe a glass knife, placing and attaching the section onto an electron microscopy grid and transferring the grid to the cryo-electron microscopy for the imaging. Okay. So, these techniques of live specimen, that is cell suspension or tissue with a thickness up to 200 micrometer and is vitrified by high pressure freezing, or that is I told that self pressurized rapid freezing technique, or directly sectioned in the cryo ultra microtome and subsequently imaged by cryo electron microscope. That CMOVIS does not necessarily involve sample purification. So, this particular technique we do not need sample purification, fixation, no dehydration is required. It is ultimate and reference technique for structural cell biology, because it is a very unique that we sample will be in hydrated condition. So, easy to go for sectioning and then image as it is. However, it is not an absolutely artifact free techniques. Definitely whenever we will do the sectioning, it may be a diamond knife or it may be a glass knife, that due to this mechanical section always there will be knife mark and the different kind of damage when it will cut the cell. In fact, the section of the vitreous biological sample is technically very difficult. So, it is not very solid and it would not be possible without a long standing effort in the microtome and diamond knife development. So, how we can use this technique? Let us take a look. So, you already know about the microtome. I discussed about this one, different parts of the microtome in details. Now, I am showing you that microtome again, where you can see there is a pipe is connected. So, here this will be much more clear, there is a hose or pipe is connected. So, this one it will be difficult to visualize because it is already blocked. So, if you look at this one, you can see this is the instrument which is connected with the hose, where it is maintaining the liquid nitrogen temperature here. So, you can see a control board here. This is the enlarged view of this same control board, where different parameters, that okay, angle of the diamond knife if you want to change it, or what is the distance, and other parameters, that if you want to fill the liquid nitrogen, all these things you can control using this control panel. Now, if you look at here, you can find out that upper control board, that is called the TC, that set the temperature in the cryo chamber. So, here there is a cryo chamber and you want to maintain the temperature. So, there is a liquid nitrogen dewar from where the liquid nitrogen supply will be there in this particular chamber, because you have to keep your sample at liquid nitrogen temperature. So, here the hose is connected to the dewar with the pump to the cryo chamber. This is the hose we are talking about. Now, we are coming to the sample holder. Here you have a sample holder that we already discussed previously, where this is the point your sample will be inserted, and here this is the knife holder, that knife will be placed here. So, this is called the knife holder, and this is knife table. So, this knife table and knife holder will place here. Here you have the knife here. This is the glass knife we can see. So, now if you go to the next slide, you can see that okay, this is the sample holder or specimen holder region, where this is the area sample will be placed, and there are different components are there which you can use this tightening the sample based on the width of that particular sample, and as well as you can position your sample, because whenever there will be a sample, if you recall previously we discussed these things, that okay, if you have a sample, I am just drawing here, that if you have a tube, generally you have a tube where your tissue or cell will be embedded. So, mostly this will be coating like that. Most of the time you have to use a blade, very sharp blade, and you have to do a sectioning of those. So, and then you have to make these things like a pencil tip, right. So, when you will do the section, your sample it will stay like that, and it will be looking like that, and this is the part we are using the glass knife or diamond knife to make the section. When you will do the section, we will get this kind of ribbon kind of thing which we are going to collect. So, this particular part it will go inside here, and this is the position we have to change of this tissue or cell in such a way it can face towards the knife, and it should not be, it will be very nicely positioned at 90-degree angle. So, there should not be any tilt between that knife as well as the sample. That adjustment we have to do it. Now, if you come to the next slide, where you can see lots of details, that when we are putting that, this is the knife, you can see this is the glass knife, this is the knife tip, you can see it, and now here there are lots of screws are there which is going to adjust that knife position tightly, that will help you to do the sectioning. So, when we are going to mount the sample and the knife, it should place in such a way so it can do the sectioning properly, and lots of adjustment you can do it within that different screw. So, there are lots of position are there that you can adjust. So, those things when you will do the practical, that part you can focus on that, what is the role of different screw and how we are tightening. The major important part is this particular area where we are using those different screw to tighten the knife, as well as we are using those screw to adjustment of this sample properly. So, when we adjusted entire sample properly, then you can see this is the sample tip here, and this is your knife tip which is going to do the sectioning. Here you can see that we are adjusting the knife properly in front of the, using those screws we are tightening the sample and changing the position. So, this is the knife, you can see it, knife tip, and this is the sample. So, we can place them properly so it can do the section properly of the tissue. Now, you can do it the same sectioning of the dehydrated sample of the epoxy resin block, or you can do the same sectioning of the tissue when it is a frozen hydrated in high pressure freezer. When the sample is completely frozen, we can use that, or we can after high pressure freezing, we can collect the cell, we can do the resin block again, and we can do this sectioning here. But here you have to remember that when you will do the cryo sectioning, continuous liquid nitrogen flow should be there. So, here you can see this is the enlarged view of this particular area where the knife and the cell, the sectioning, it is happening. If we go to the next slide, it will be much more clear here. In this particular case, you can see it that we have a grid. So, if you look at here, we have a grid here, and this is the cell. As I mentioned that when there is a cell and blade is constantly doing the section, so what will happen? It will make a small kind of ribbon. So, how this ribbon will look like? So, last section I was showing you. So, if this is our sample or block, if we just consider, and this is my tissue is embedded, and this is my glass knife, it is going to do the section. So, this is the top part of the glass knife, and it will continuously. So, this is the sample, this is the, if you just consider this is the sample, and the glass knife, it will constantly go up and down with this sample, and then what will happen, a very fine section of this tissue it will come out, or tissue with the resin it will come out, and it will start floating there. So, when the knife, it will constantly go up and down, and the tissue will also move gradually towards this direction, and it will make a small ribbon like thing. So, when this section it will come, what will happen, we will get this kind of small ribbon will top of each other. So, that small ribbon we are going to collect in this grid. Here you can see this is the sample area, and this is the grid, we are collecting that sample. So, this is the ribbon, you can see there is a eyelash is there, which is helping to put that ribbon on top of the grid. So, we are using this eyelash to bring that ribbon on top of that grid. So, here you can see the ribbon is there. So, if we are doing entire process in presence of liquid nitrogen, immediately grid should be placed in the grid box. After placing that ribbon on top of this, and entire exercise should be done at liquid nitrogen condition. So, here we are showing you in the entire view how it is looking. So, when the section is done using this glass knife, we are using the grid and trying to collect that small section on top of the grid, and that we are using for the imaging purposes using either cryogenic condition or in a dry room temperature condition. It is up to us that what condition we are going to image this entire sample. Okay. So, next, that is called the fridge fracture, that we discussed about that couple of times, that when we started this discussion, today's discussion, that we are going to use cryo ultra microtome for the sectioning of the sample. So, fridge fracture is the another technique where we are going to disrupt the biomembrane, and that is the plasma membrane. So, which part of the cell it is going to rupture, that is basically the hydrophobic core region. First we will freeze the sample, when we will freeze the sample, biomembrane it will be rigid, and then either we will use a glass knife to break the cell. So, if this is our cell, we will just use the glass knife to rupture the cell membrane here. So, we are trying to make it separate the cell membrane in two parts. So, that is the technique we are calling that, this is called the fridge fracture. Now, take a look how this fridge fracture it is working. So, that biological macromolecule, especially the cell is rapidly frozen to immobilize its cellular components. So, if you freeze it rapidly, most of the cellular components will be frozen, and then it will be immobilized. This freezing is performed in the presence of cryoprotective reagents such as glycerol, which help to prevent the formation of the ice crystal. The frozen sample, so here we are not using any high pressure technique. So, we are just trying to avoid that high pressure technique. So, either it may be a mammalian cell or it may be a insect cell or it may be a bacterial cell. When we have those, we are using a glycerol, because glycerol will behave like a cryoprotectant. So, we will mix the cell with a glycerol and then we will freeze that entire cell. Then frozen sample is fractured under using a different way, like high vacuum at liquid nitrogen temperature or using a knife. This fracture usually happen near the natural lines where cell or organelles membrane are weak, like plasma membrane or outer membrane of the organelles, creating two distinct phases, that is the PF phase, that is plasma fractured phase, and the EF phase, that is called extracellular fracture phase. So, that I will show you when I have some of those images and diagram. So, following the fracture, the exposed surface are shadowed with the platinum vapor at an analog to add 3-dimensional details to the surface. If you remember, when we are talking about the staining of DNA, that time I discussed about the metal coating or we are using the platinum to coat the DNA. So, one side of the DNA will be coated and other side there will be no coating, and then there will be a very nice contrast difference and you can see the entire DNA properly. So, that was the idea for that DNA sample. Why we have to do it? Because there is a carbon coating, and if there is a DNA is there, if there is a no metal shadow or carbon shadow, it will be very difficult to distinguish the DNA from the carbon background. That is the reason we are doing that metal shadow. Here also, when we rupture the cell, first we freeze that cell and then we are using that some kind of knife to rupture the membrane. When membrane is ruptured, so there will be not enough contrast difference, so you can see inside the cell. In this particular case, what is required? You need a platinum vapor or this metal shadowing technique, this shadowing technique to coat one side of that fractured cell. So, if the cell is fractured in one particular side, so it will be one side, it will be completely exposed and the metal it will deposit in that particular area. So, then there will be a contrast difference between the one side and the other side, and if you look at through the rupture part, you can see the inner part of the cell. So, it is basically increasing the contrast of those. After that, the carbon is then evaporated onto the sample vertically, forming a carbon metal replica of the fractured surface. After the organic material is digested away, the resulting carbon replica is mounted onto an electron microscope grid for the details examination. So, this is the procedure of the fridge fracture. So, fridge fracture methods provides a unique representation of the structural and functional element of the cell membrane. So, cell membrane is staying as it is, we are just freezing the cell membrane and then mechanically trying to rupture, and if you look at the cell membrane, we can see lots of details and elements of the cell membrane. The electron beam used in the TEM passes through the carbon portion of the replica while being absorbed by the platinum area, produce a high resolution 3-dimensional images. So, these images allow scientists to study the detailed about the cell membrane in a way that traditional section method cannot do it. That is the reason this fridge fracture is getting very popular among the different cell biologists. So, scientists are using these things very frequently. One of the critical application of the fridge fracture technique is its ability to visualize the arrangement of the protein and other molecules within the membrane, contributing to our understanding of the membrane function. Additionally, this method is used to various area of the cell biology, nanotechnology, offering insight into the molecular structure and material sciences. How it is working? So, here you can see this is the instrument where you have the turbomolecular pump, because you need a high vacuum here. Why do you need high vacuum? Because we are talking about that platinum coating or the carbon coating. Platinum coating and carbon coating, you have to do it inside a coater machine, maybe a carbon coater or the metal coater, and you need a very high vacuum. If you do not have the high vacuum, metal cannot spread uniformly. It will hit after the collision with the air molecule, it will distribute in a different way. So, it will not distribute it properly. So, that is the reason turbomolecular pump is there, which will maintain the temperature. Cold shroud is there, which is going to do the rupture the membrane, and temperature controlled stage is here, where we have the stage where it is maintaining the very low temperature, and this is the knife or the microtome blade which is going to do the section. So, cell will be here at the top where there is a temperature is controlled and very low temperature, and this blade is going to do the section. Here port for the specimen entry, through this one we will put the specimen here, and then this is the carbon electron gun and the platinum electron gun. After the fridge fracture, we are using this gun, the carbon and platinum, to coat with the platinum as well as the carbon. So, that I already discussed. How we are doing this fracture? Here there is a different types we can apply. So, I already talked about the knife. We are using those cell is already frozen, and this is the blade at the cold temperature. We are using and making that fracture or cutting that cell, and then we are using this one to put it on the grid to visualize. Or the tensile fracture also we can use it, where the cell is attached and then at cold temperature we are putting it and then taking it away. So, when the cell is completely frozen, if we just give some pressure, cell rupture, it will happen. So, after this you can see when the cell rupture it is happening, basically what is happening, there is a extracellular membrane. This extracellular membrane, it will be separated out from each other, and there will be a gap between when the membrane is ruptured. So, it is like that we have a shot, we are just ruptured the shot completely and then trying to look inside the cell. So, here also forcefully we are rupturing that extracellular membrane where some proteins are embedded. So, if you tear our shot, so what will happen, sometime the button and other things it will be visible there. So, exactly same thing when we rupture this cell membrane, what we can see intercellular those protein molecules or membrane protein, as well as we can see inside the cell. So, here there are some results we can see it, where you can see inside of the cytoplasmic layer and inside of the extracellular layer, where if you focus on this particular area you can see lot of organelles, cell vesicles, membrane, how they are interacting or gradually they are changing their position, or from that membrane how it is forming these extracellular vesicles from here. So, that is a very interesting technique that we can implement. Those are the equipments we can use it for the fridge fracture, that okay. So, we need definitely liquid nitrogen or propane for fridging system. So, fridging machinery or fridge fracture machine is required, fracturing control the fracturing of the frozen sample. Vacuum chamber it is required, because as I mentioned, for the metal coating or carbon coating we need that kind of vacuum chamber, that sputter coater or the vacuum evaporator. Then transmission electron microscope, sample mounting device, those are the normal for any cryo-EM instrument. Now, we are coming back to the FIB-ACM. So, FIB-ACM, as I mentioned, that it is called focused ion beam. So, this is a dual beam system. Previously we can use the focused beam to damage the cell, but nowadays we are using that focused ion beam and scanning electron microscope. Using the scanning electron microscope we are studying the surface of the cell, and then using the focused ion beam to rupture the cell membrane. So, what we are doing, the first instrument was the single beam, not the dual beam. It was the ion beam microscope, and later it was combined with the electron column, and that is the point where the scanning electron microscope it is coming. This ion beam is placed at an angle, so 38 or 36 degree to the vertical of the electron beam. So, electron beam it will come like that. So, we look at the surface, and then the focused ion beam, it is mostly gallium, it is coming in a certain angle, and that angle is 38 degree or 36 degree, and that is basically focusing in basically same place. So, coincide at a working distance of about 4 millimeter. So, where we are focusing one particular area, and then that focused ion beam will going to focus in one particular area and rupture that cell membrane. Since the focused Fib SEM technology entered in that biological application about 10 years ago, it gained its merit in preparing that TEM and cryo-EM sample. For the material that is difficult to cut, we cannot use cryo ultramicrotome either by the hardness or the large difference in the hardness or incompatibility with diamond knife. So, those are the cases Fib SEM is really helpful, and also after this sectioning it is not going to keep any knife mark on your sample. That is the another good thing, that when we want to use that Fib SEM. So, either this ion beam can be used to cross section of the cell or organisms prepared for sectioning for scanning electron microscope to access the internal microstructure or study the cell substrate interaction. The most promising technology at this moment, however, is the Fib SEM tomography from a sample embedded in resin. A cutting plane from the surface of the resin block is opened and pre-selected region of interest we are going to focus. So, that I am going to show you in the next slide. So, if you are considering that resin block, so most important part is that when we have a block or we have a tissue, so that will be embedded in the resin, or there is a plastification on top of the cell, and then first we have to focus on the region of interest. When you will find the region of interest, then we have to identify that particular area within this large resin block, and when we will identify that region of interest, then we are going to use that the ion beam to precisely locate that area and use that ion beam to mill that membrane and see inside the cell. So, here you can see this is the resin block. So, resin block you have to cut that entire resin, and then you have to reach near the cell, and then you have to do that sectioning in such a way that it should not damage the cell. But if you just use that plastification, that the thin layer plastification, here you can see this is the cell membrane on top, and then we are doing a thin layer plastification that will decrease the depth, and it will be easy for the focused ion beam to rupture that outer cell membrane easily. That is one of the reason we are preferring that plastification on top of the cell before we cut the cell at different angle. So, I am not going through all the details about those techniques because of interest of time, we do not have enough time to go in details. You can go through the book, if you are interested we can discuss more. So, here you can see those are the cell after the plastification, and then we are couple of area we selected, we mark those area for the sectioning, that this is the interest of our cell, and then we can do the sectioning and we can see lots of details inside that cell. So, here that is we are talking about that there will be one electron beam from the scanning electron microscope, it will come like that, and the gallium ion beam it will come from a certain angle to rupture the cell membrane, and that will help us to to visualize inside the cell. So, I will directly go to one example where you can see it. Here you can see this is our SEM. We are targeting particular this area. This is my interest area that I want to focus this particular area. Then I am using that 50° tilt fib, that focused ion beam. So, this is our 50° tilt from the SEM image. This is the target area. Then I am using the 50° tilt fib image, and we are trying to open, forming a small window here. So, in this enlarged view you can see this membrane is completely ruptured, which is deposited here. We are milling with that focused ion beam and gradually milling this entire part and open this entire area. Now, we are focused on this particular area for the imaging using the SEM, where you can see this is the outer part where this is visible here, and you can see lots of other cell organelles in this particular region. So, using this one we are not damaging the cell too much, we just focused on that particular region and see all the details. Now, you can see all the details of that particular cell. You can see lots of mitochondria there, as well as you can see lot of extracellular vesicle and other organelles here. You can also use that SEM to calculate the 3D view of this entire cell inside the cell and get the 3-dimensional structure. Same way, I will just quickly go through this correlative light and electron microscope. We are tagging sometime. I will just go to the example part, it will be easier. Sometime cell we are tagging with a fluorescence tag, and then we are using the cell slide in a fluorescence microscope. We identify that particular area where our protein of interest. If you look at this particular cell, you can see those are the fluorescence label tag. It is a red fluorescence label tag or green fluorescence label tag. Where there is a red and green is mixture, we are getting some yellow tag. Now, if you just consider that this is my area of interest, this particular area of interest in same orientation will insert in cryo-EM, and then or we are taking this particular area, we are using the fib SEM to rupture the cell membrane and inserting that one for using that SEM or cryo-EM to image inside that particular area. So, if you just consider that okay, my protein is fluorescently tagged, it may be green, red or yellow. So, we will just focus on that. We because of that fluorescence tag, we will just use that FIB focused ion beam, will just rupture the membrane, and we can focus on those particular protein at high resolution imaging technique using the cryo-EM or cryo-TEM. So, at this moment we will stop here. About all this cell membrane rupture and doing the cell sectioning. We will talk little bit more about how to use graphene or graphene oxide to prepare the biological science sample in the next class, and then we will discuss more about the 3D reconstruction or data collection techniques. Thank you all for your attention.